Okay, let's start. In the last class, we finished most of reproduction and inheritance. The only thing left is gene regulation and biotechnology. We did the diseases associated with autosomal and linked to chromosome X. We also did all of this, the DNA, prokaryotic DNA. We did structure and replication of DNA. We did all of that. So today we'll do biotechnology and gene regulation. So this was me doing uh, all of the stuff earlier. So today we'll focus on gene regulation. What is gene expression? We did gene expression in the last class. Gene expression is the process where where gene is being transcribed to mRNA and then translated to protein. So gene expression is you have transcription, and translation. And gene regulation is what we'll do today. And gene regulation is the process that enables gene expression. So without gene regulation, you cannot have gene expression. So there are two types of genes that you need to know: the structural genes and the regulatory genes. The structural genes are the genes that code for protein needed for cell structure. So it says what its structural genes basically codes for proteins and uh, proteins that are needed for cell structure and function. So basically, collagen is one of the proteins. So the structural genes will code for collagen and keratin as well, and a lot of other examples. Keratin, yeah. So what are regulatory genes? Regulatory genes are genes that code for regulatory proteins that control gene expression or transcription. And remember that regulatory genes are involved in gene regulation. And the type of regulatory gene are called transcription factors. So a transcription factor can bind to a specific DNA sequence and control transcription. So a transcription factor are basically proteins. They are proteins that are made by regulatory genes, that are coded by regulatory genes. And these transcription factors can bind to a specific DNA sequence. So the specific DNA sequence that they can bind, the place for binding are of two types. So you have the activators, which increase gene expression, and you have the repressor, which inhibit gene expression. So repressor proteins, they would inhibit gene expression, and activators are the proteins that increase gene expression. So and repressible enzymes. Repressible enzymes are their synthesis can be prevented when a repressor protein binds to an operator, a specific sequence of DNA. So remember, repressible enzyme is a repressor protein, and it binds to a place called operator. Operator is a specific sequence of DNA. So this is if this is the operator, then the repressible enzyme can bind to this operator, and so it would block the DNA polymerase. This is DNA polymerase from moving forward. So DNA polymerase cannot move forward, and therefore transcription cannot happen. And so repressor stops binding of RNA polymerase. So RNA or DNA polymerase, because DNA polymerase cannot move forward because of this repressible enzyme or repressor protein. So this stops DNA transcription. And remember, there is something called inducible enzymes as well. Uh, this causes synthesis only when substrate is present. And inducers that bind to regulatory proteins gene is switched on. So inducible enzymes can only work when you have a substrate. We'll talk more about this later on, but for now, just know inducible enzymes only work when you have a substrate. If you don't have a substrate, then inducible enzyme won't work. And there is gene control in prokaryotes that you need to know. This is the most important topic. Gene control in prokaryotes. So this is found in the prokaryotic DNA. Regulatory gene is close to something called the operon. operon. Remember this name. What is an operon? An operon consists of one or more structural genes. So the genes that code for regular proteins for structure. And this is under control of a single promoter. And promoter is the place where RNA polymerase binds. Remember, control region of DNA, which is specific sequence recognized by regulatory protein and enzymes. So promoter and operator are two regions of in DNA sequence where the proteins can bind to. So promoter's job is to bind RNA polymerase to, and promoter, from its name, promotes transcription and translation. But the operator's job, operator is where the repressor protein binds. So an operator stops DNA transcription. So promoter promotes, operator stops. And so this is for, you can't, I don't think you can see it, but let me try. So this is the DNA sequence. For example, this is the DNA sequence, and this is the place where the regulatory genes are present. And you'll always have promoter before the operator. So remember, promoter comes before the operator. And remember that the operator is where the repressor protein binds. So this repressor protein comes and binds to the operator. So now you have the repressor protein bounded to the operator. And now RNA polymerase can bind to the promoter, but it cannot move forward because you have a repressor protein in front. So because of this, your RNA and DNA transcription and translation would stop. So why am I explaining this? Because this will cause your structural genes from being coded. So RNA polymerase will stop the transcription of structural genes. So structural genes cannot be transcribed and translated, so you won't have structural proteins. So the reason you have an operator is so that structural genes cannot be coded. So yeah. And so let's move on to an example. This you have to know. It's called the lac operon. A lac operon is a part of DNA which is found in bacteria. And bacteria, remember, it lies on sugar, glucose, and lactose. Remember, bacteria would need sugar, glucose, and lactose for growth. And what is lac operon? It is a set of 3 genes next to each other. And these 3 genes, remember, they code for 3 repressible inducible enzymes. The 3 genes are LAC-Z, which codes for beta-galactosidase, LAC-Y, which codes for permease, and LAC-A, which codes for transcriptase. I don't know how to pronounce this. I still can't pronounce this. So you have to memorize what the role of each gene is. So LAC-Z, LAC-Y, LAC-A, you have to know what it codes for. So yeah, and these 3 genes, remember, these 3 genes, they would break down lactose. The job of these 3 genes are to break down lactose. So all of these 3 enzymes that they code for, so each enzyme has a role in breaking down lactose. So this is lac operon. This is the DNA sequence of lac operon. So you have the promoter first. You have the promoter here. This is the promoter where RNA polymerase would bind. And this is the operator. This is the operator where the repressor protein would bind. And if you have the repressor protein bound, then DNA polymerase cannot move forward. So therefore, these 3 genes cannot be transcribed. So with the lac operon, there is a regulatory gene. Remember, there is a regulatory gene called LAC-I. This LAC-I regulatory gene codes for a regulatory protein. And this, so regulatory gene LAC-I codes for a repressor protein that is close by. And repressor is a constitutive protein. remember, this repressor protein that this regulatory gene codes for is a constitutive protein. Constitutive protein means that it is produced at all times. No matter if transcription is happening or not, this regulatory gene will always code for this regulatory protein. So this repressor protein that is being made here is made at all times. Whether you'll have transcription or not, it doesn't matter, it will still be produced. But there is a problem. This repressor protein is an allosteric protein. This means that this repressor protein has two binding sites. This repressor protein has two binding sites. So it's shaped something like this. So this is one binding site. This is the second binding site. And the first binding site, this first binding site is where the operator would bind to. So this is the binding site where the operator binds. And this site right here, the second binding site, is the site for where the lactose would bind. And this lactose, when this lactose binds, the protein would change shape. So what is happening? When lactose binds to the site, protein changes shape. So if you have lactose present and it binds to, if you have lactose present, then the lactose would bind to this regulatory protein, and this protein would change shape. So when this changes shape, so you have this lactose bounded to the protein, then the protein changes shape, and this protein can no longer bind to the operator. So since the binding site of the operator is changed now, you cannot bind operator to this repressor protein. So when lactose binds to the protein, the DNA binding site or the operator binding site is closed, and the repressor protein is now inactivated. So what does inactivated mean? Inactivated basically means that the repressor protein cannot bind to the operator. Yeah, I wrote the wrong spelling. Never mind. So yeah, this is how basically this lac operon works. If you have lactose present, the repressor protein changes shape, and you can no longer bind to the operator. So why is this important? This is important because if you have lactose present, right? If you have lactose present, then the repressor proteins are no longer activated. So when you have lactose present, the repressor protein are no longer activated. So the three enzymes, the lac A, lac Y, and lac Z, they can be coded for. So these three enzymes can be coded for, and then these three enzymes can break down lactose. So remember, the job of all of lac operon is so that repressor proteins are no longer activated. So when repressor genes and proteins are no longer activated, these three genes can be coded for. So when these three genes are coded, they can break down lactose. So yeah, so what happens when you don't have lactose? When you don't have lactose present, the regulatory gene would code for the repressor protein as it usually does, and the repressor is not bound to lactose. So the repressor protein is not bounded to lactose. This means that the operator can be bounded to the repressor protein. So the repressor binds to the operator region, because remember, you don't have lactose, so the binding site does not change shape. And so the operator is now bound to the repressor protein, and RNA polymerase can't move to the operator, and structural genes. So no transcription of all these three genes. So the genes are switched off. So the genes are switched off. So regulatory genes are coded for. So these prevent the transcription of these three genes. So when you don't have lactose, the repressor genes are switched on, and these repressor genes prevent the translation and transcription for these three structural genes. So now these genes are not produced, and so since there is no lactose, then you don't need these genes to break down any lactose. So no breakdown will happen because you don't have lactose, so you don't need these three genes. But again, when lactose is present, lactose acts as something called the inducer and binds to the repressor protein. Repressor protein changes shape and can't bind to the operator and block genes. So it can't bind to the operator region, so you can't block the genes. So transcription happens and RNA polymerase binds to the promoter. So RNA polymerase binds to the promoter and it can move forward, and transcription happens. So yeah, this was lac operon. So you have to know all of this. And the last thing for today, so that we can finish this chapter, is genetic engineering and biotechnology. So the first thing in biotechnology is PCR, polymerase chain reaction. You don't need to know the name, just know the short form. So what is PCR and why is it needed? PCR basically clones and amplifies DNA. So that you can use DNA for many other purposes. We'll learn about that. But for now, the job and role of PCR is to clone and amplify DNA. And it is a rapid and efficient process. So what are the components that you need? First of all, you need template DNA. Second, you need a Taq polymerase. What is Taq polymerase? It is a DNA polymerase that is heat stable, and it is acquired from aquatic bacteria, the bacteria that live in water. Taq polymerase is taken from here. And you also need a buffer solution. A buffer solution basically someone has a question. Are acetylation and methylation not required in gene expression? Acetylation and methylation, what? No, I don't think this, what I just explained was gene regulation, not gene expression. And I've not heard about acetylation and methylation in both A levels and IGCSE. So I don't know what you're talking about. But so let's move on with buffers. Buffers, they control the pH. You want a stable pH so that the hydrogen bonds of DNA doesn't break. Because remember that if the pH becomes too low, then the hydrogen bonds can break. Because low pH means you have high H plus concentration. This can affect both the ionic as well as the hydrogen bonding. So pH can result in these two changes, ionic changes and hydrogen bond changes. So yeah, this is why buffer is used. A buffer contains KCl and MgCl2. So remember what buffers are made up of. And you also need, apart from buffers, you also need the 4 different nucleotides. So you need adenine, thymine, you need cytosine and guanine. And you also need 2 primers. A primer is a short sequence of single-stranded DNA or RNA. So yeah, and how does PCR work? So the first step in PCR is denaturation. It's when you denature the DNA strand, you basically separate the 2 DNA strands. So initially, if this was the DNA strand, now you would separate the DNA strands into 2 single strands. And this happens at 95 degrees. Remember, you have to know the temperature of each step. And so this is the first step. DNA strands are broken into 2 separate strands. The hydrogen bonds between the DNA strands break. Why does this break? Because remember, this happens due to temperature changes, not pH changes. So temperature change causes hydrogen bonds to break. And so when this breaks, the bases on the DNA strand are now exposed. So these bases are exposed, and the second step comes into play. So the step 2 of PCR is annealing, and this is when the primer is added. So the role of primer is that it binds to the target region of DNA or single-stranded DNA for amplification. So it acts as a starting point for Taq polymerase, or this is RNA or DNA polymerase to bind. So Taq polymerase binds to the primer, and then transcription happens. So Taq polymerase only binds to the double-stranded DNA and adds new nucleotides to the existing strand. So the primer, when this primer is added, the Taq polymerase can bind to this primer and start transcription of this DNA. So both primers are added. You need 2 primers in PCR. So both of these primers will act as the place for Taq polymerase to bind and start transcription. So primer are added and new hydrogen bonds start to form because Taq polymerase hydrolyzes new hydrogen bonds. So this is step 2. And finally, step 3 is extension. And yeah, remember that annealing happens at around 60 to 65 degrees. So 60 to 65 degrees, remember this. And extension is when Taq polymerase binds to the primer and synthesizes new DNA strands. At step 2, the primer was added, and in step 3, the Taq polymerase binds to the primer. So the new DNA strands are synthesized and complementary. The DNA strands are complementary to the DNA template strands. So extension, remember, happens at 70 to 75 degrees Celsius. Now, the reason you use Taq polymerase is because this protein is very heat stable. It can withstand high temperatures. It is very heat stable. So usually if it was a normal polymerase, it would start to denature at around 50 degrees. That's the temperature at which enzymes start to denature. So because you use Taq polymerase, the Taq polymerase would not denature, and because of this, you don't need to replace the DNA, the Taq polymerase every time you do PCR. So Taq polymerase is very heat stable, and the reason you use it is because you don't need to replace it. So the PCR becomes cheap. Because if you wanted to replace Taq polymerase every time, then you'd have to make new enzymes. This will cause money. So yeah, this is the reason you use Taq polymerase. And DNA is heated again to separate the strands. So when you make the new DNA strands, this is heated again, it's separated again, and the whole process is repeated. So you repeat the whole extension, you repeat the 3 basic steps, and you make more DNA until you have enough DNA required. So the number of DNA doubles every PCR cycle. So this is an efficient process. So the number of DNA doubles. So the DNA copies of each PCR made is 2n. So this is the number of PCR you did. And what are the advantages and disadvantages of PCR? As I said, it is rapid and efficient, and you only need a small sample of DNA to start amplification. Amplification is basically when you amplify DNA, you make more DNA. And the process is all automated in a thermal cycler. So you don't need any workers for this. But the disadvantage is that you need to know the precise DNA sequence beforehand, because remember that primers must be complementary to the DNA template. And so for this reason, the primers must already be created before the DNA strand, and you need to know the precise DNA sequence for this DNA template. So this causes a lot of problems, because finding the precise DNA sequence is not an easy thing. And what are the applications for PCR? Remember, you can use PCR for DNA sequencing. you can also use it for genetic screening, and you can use it for recombinant DNA technology. This is what we'll do next. But yeah, DNA sequencing is, you remember the PCR test, the Corona PCR test? This is one of its applications. So yeah, 3 applications, know them. And so moving on to recombinant DNA technology. This is important if you have misused, or you have problems in your genes. So what happens is DNA is recombined with plasmid and then sent back into the bacteria. And so new genes are produced that are working perfectly fine. So how does recombinant DNA technology work? There are 6 steps. The first step is you identify and isolate the mRNA or DNA from the organism. This mRNA or DNA must be working. And step 2 is you cut the target gene and plasmid DNA. So first step was you isolate the mRNA or DNA sequence from the organism. Step 2 is you cut the target gene, the gene that you need from this DNA sequence, and you also cut a part you cut DNA plasmid. And then step 3 is you recombine this plasmid DNA with this target gene. So these two combine. And step 4 is you insert this recombinant plasmid. So the newly made plasmid, the plasmid that consists of this target gene, is inserted into the bacteria. And step 3 is you identify the transformed bacteria. So when the plasmid is inserted into the bacteria would produce these genes, and you then identify the transformed bacteria, and you grow this bacteria in fermenter for large scale gene production. So you need to know the application and the procedure as well in detail. One of its applications is when you need human insulin. So when you don't have human insulin, right, you'll have diabetes. When the body doesn't produce enough human insulin, you'll have diabetes. And to prevent this, to make insulin, humans use recombinant DNA technology. So how is insulin produced? Step 1 is you obtain mRNA for human insulin. Now this mRNA must be working. This because remember, you can't take genes cannot be taken from person that has diabetes. So you need to take mRNA from a healthy human being that produces enough insulin. So this can be taken from the beta cells of isolates of Langerhans in pancreas, and then you reverse transcriptase to make complementary DNA from mRNA single-stranded, and then DNA polymerase is used to make double-stranded complementary DNA. And so the final result is the genes with that are short and that doesn't have any introns. We'll learn about introns more, but yeah, for now, first step is to isolate the mRNA and make enough mRNA or DNA. So DNA is amplified using PCR. This is where the PCR will come into play. You need enough DNA for human insulin to be produced in bacteria. So PCR is used to amplify the DNA. Which DNA? The genes that code for human insulin. So this was step 1. Step 2 is you cut the target gene and plasmid DNA. So when you have the DNA produced, enough DNA produced, you cut the specific gene that produces insulin. And you also obtain plasmid from the bacteria and cut plasmid at a restriction site using enzyme. Now, the reason that you cut this plasmid, so this is a circular plasmid, is because you can, so that you can insert the human insulin gene into this plasmid. So yeah, and all of this cutting happens because of an enzyme called the restriction enzyme. So a restriction enzyme is from a bacteria. It recognizes, binds, and cuts DNA sequence. Uh, and it's the sequence or the place where the restriction site cuts is called the restriction site. So the place where the cut happens is called the restriction site. And it also hydrolyzes the phosphodiester bonds between nucleotides. So when the human insulin gene is combined with the plasmid, this restriction enzyme would hydrolyze the phosphodiester bonds between the nucleotides. And so this enzyme also produces sticky ends. Sticky ends are result of a staggered cut. And at the sticky ends, you will have unpaired nucleotides at the ends. So this means that the hydrogen bonds can easily be formed. Does restriction enzymes only hydrolyzes phosphodiester bonds? What about hydrogen bonds? It also forms hydrogen bonds. Remember, sticky ends are formed because so that you can form hydrogen bonds. So restriction enzymes produces sticky ends. Sticky ends have unpaired nucleotides. And because of these unpaired nucleotides, you can form hydrogen bonds between these nucleotides. But if you have a blunt end basically means that you have no unpaired nucleotides. So yeah, restriction enzymes produces sticky ends. And what is step 3? Step 3 is when you recombine this human insulin gene to the plasmid DNA. So the human gene is mixed with the plasmid, and DNA ligase seals any nicks in sugar phosphate backbone. And it also catalyzes the formation of phosphodiester bonds to form recombinant DNA. So remember the enzymes that are needed for DNA, recombinant DNA technology. You first of all, you need Taq polymerase for Taq polymerase. This is not specifically in the recombinant DNA technology, but you need this for PCR. And remember, recombinant DNA technology always uses PCR because you need a lot of DNA for to produce a lot of human insulin. So first, Taq polymerase. Second, the second enzyme is the restriction enzyme. This is needed to cut and bind the human gene to the plasmid of bacteria. And the third enzyme is DNA ligase. And DNA ligase, remember, it would seal nicks in sugar phosphate backbone. And it also catalyzes the formation of phosphodiester bonds. So yeah, step 4 is when you insert this recombinant plasmid into the bacteria. And so the recombinant plasmid is mixed with the bacteria, and you treat this bacteria with a solution of Ca2 plus ions, and you allow this to cool. Why does this happen? Because the plasmid is not inserted into the bacteria directly. You need to apply heat shock or use electrophoration to increase the chances of plasmid passing through the cell surface membrane. Remember that plasmids need to pass through the cell surface membrane, and this cell surface membrane is partially permeable. So yeah, without this heat shock and Ca2 plus ions, the plasmid cannot enter the bacteria. So when this happens, when the plasmid enters the bacteria, step 5 is you identify the modified bacteria. How do you identify the bacteria which will produce this new genes, which will code for these newly plasmid? You identify them by using marker genes. Marker genes are placed in plasmid to help recognize the recombinant plasmid or the recombinant bacteria. So genes of interest is added close to the marker genes. And so when the recombinant bacteria is produced, you can identify them by marker genes. And you can use a number of different ways to identify transformed bacteria. You can use antibiotic selection, You can use fluorescent proteins. So insert fluorescent proteins into the bacteria, into the recombinant bacteria. So when you place the bacteria under UV light, the bacteria would fluorescent, or it would glow. Or you can use any easily stained substance and attach it to the bacteria. For example, beta galactosidase. And these are the three ways you can identify transformed bacteria. And there was step 6. So step 6 was that you use the recombinant bacteria. So the bacteria that is modified through the modified plasmid, you use this bacteria, you place it in a heat fermenter, and you reproduce this bacteria. And so you reproduce this bacteria, and because of this, insulin can be made. So yeah, this is step 6. And so you have to know all of these 6 steps. And you also need to know why plasmids are used for bacteria. Why do you insert the human insulin gene into the plasmid of the bacteria? Because plasmids are small, so they can be easily inserted into bacteria cells. They are circular, so that means they are more stable. And plasmids can act as vectors to deliver human genes into bacteria. And another reason is that it can easily be extracted from bacteria, and it can be taken up easily by the bacteria. And so yeah, these are the advantages of plasmid, using plasmids. And yeah, with this, we have finished biotechnology. And also with this, we have finished this chapter. So yeah, all of this we did finish. And so yeah, we only have 2 chapters remaining. So we'll do that in the next classes. So yeah, bye. See you in the next class.